Good morning, all. I hope you can all hear me fine. I'm Francesca Chianini, as you may be aware, and I'm a vet pathologist at the Moredun Institute. So I'm speaking to you from Scotland. It's my pleasure to give this presentation to you today. And before starting, I want to thank Abuja University and blessing Dr. Sande for inviting me to give this talk. And also just another, well, very, very brief presentation of my background to explain like my career so far. So I started my career as a vet. I studied as a vet and I graduated in Italy in Pisa University. And then I moved for a master to Barcelona to the UAB University. I've done a master on lymphomas of lymphoma in the brain of cats. And then I was given a grant, an European grant, to carry out a PhD on a disease of pigs, the circovirus in pigs. And as soon as I finished my PhD, I joined Moredun, where I've been working as a veterinary research pathologist for over 18 years. I'm currently not strictly a research pathologist. I'm more a diagnostic pathologist. I work in the surveillance, and the presentation I'm going to give you today is more about the diagnosis of neurological condition in ruminants. And also, before I go ahead, I just warn you that you won't be able to join me with a live question, but we have an A and Q. I think you can maybe see it in the screen. So you have the description on how to write a question if you have any. I won't be answering the question live, so I'll answer some of the question. Hopefully there are some at the end, and I'll possibly select some. But if I don't have time to finish to address all the question, don't worry, because there will be a Q&A report at the end of the event. Also, I just want to make you aware that if you have any problem, like technical problem, bear in mind this is my very first time to give a live event with this platform. Fortunately, I have a great assistant here with me, but you know, things sometimes can go wrong, and that's why we decided to record the presentation. And after that, we'll circulate a link where you can access the recorded presentation in the future. So I think I can now start my presentation. Okay. Great. So I hope you can hear me. That's again, as you know, I'm gonna talk to you about the neuropathology of ruminants. Even before start, I want to say that, of course, my presentation is gonna be about a limited number of diseases. If I had, you know, like to explain the all the large range of diseases, even just in ruminants, that will take a very, very long time. So I concentrated mainly on the pathological neuropathology disease, that are present in the UK, so that I can even see in Scotland during my experience as a diagnostician. So the brain is quite a special organ. Of course, all the organs are special and important, but the brain is somehow like different. And this is also possibly why neuropathologists are almost a niche group of pathologists, because even like very talented histopathologists sometimes are a bit like almost scared by this organ, which is not the case because it's the most wonderful and fantastic organ. Nevertheless, it's pretty complicated to like get a good grasp of not just the disease, but also of the variety of the anatomy that exists between animals. So I just put up a slide taken from the internet, just to show the variability of size between animals, so like a whale to a small rodent, and like even like how comparatively a brain of an adult sheep compared to a deer. So first of all, you know, like just to make you aware that there is a huge variability. Of course, there is variability between other organs like lung and livers, but the variability most of the time and just like generally for other organs is more macroscopic than microscopic. But the brain has a very special like anatomic difference and histopathology difference, because each area is somehow specialized. So you cannot examine just a tiny bit of brain and get to a diagnosis, whereas most of the time you can, if the disease is spread out, of course, with almost whatever bits and like piece of other organ, you may have a good chance of spotting the lesions there. So like, let's start from the from how we approach the study of neuropathology. So the first important thing, like any other organ, is macroscopic examination. So you have to be familiar with the brain. That's why I showed the normal and size brain and shape. You have to be familiar with the brain, how it looks like. In this slide, you see like an example of a brain just extracted from a sheep from like when I was doing research in prion diseases. So when the brain is extracted, this is a fresh brain, you can see some trace of blood, so the cerebellum, the cerebrum, and here even a tiny bit of spinal cord. So once you've extracted the brain, you can check it, and sometimes you can do it when the brain is fresh, if the macroscopic alter feature is really clear. But most of the time, what you'll do, you fix the whole brain in a large amount of formalin, so to allow it to get not as soft as it is when you extract it from the skull, and then you can trim it and check if there are any alter pattern. So as you see here, you have from the macroscopic examination, you can have deviation of normal anatomy. This is, as I said, sometimes you see it very clearly. We'll see some picture later on. Otherwise, there are space-occupying mass. Again, sometimes you see them already without cutting the brain or anything. Large abscesses, again, we'll have some example, and you see them macroscopically. Some other time is a bit more difficult, depending on the level of the alteration of the tissue, but you may see some like altered white matter, so the center, the core of the brain. So you can see some pallor, the consistency is softer, and usually this is due to myelin disorder. And another altered pattern you can see macroscopically is malation necrosis. This I think I have some example of that too. They can be due to a decently long list of cause. So like trauma, infert, or metabolic, or even toxic problem, or mitochondrial disease. I won't be able to touch on all these changes, but I'll try to illustrate the more important one to you. And then we have, of course, what I do it almost constantly on my day-by-day work, sitting at the microscope, usually like a larger one than the one you see here, that allows me to see like what's going on deep down in the tissue. Just to make it as, you know, like as immediate as possible, I separated which kind of feature, what you can see. Of course, this is not exhaustive, it's just like to give you a flavor of what you see down the microscope. So the main feature that you can only see down the microscope are perivascular caffeine and glial foci. Now, some can claim that there are some occasions where also abscesses, so infiltration of pus, is only observable macroscopically. Like in some cases, and again, we'll have some cases where we see it, where the abscesses are really micro abscesses. So the only way to see them is to spot them down the microscope. But certainly perivascular caffeine and glial foci, you can only see them down the microscope. Usually they are they reflect, they are what is observed in case of inflammation due to infectious diseases or immune diseases. Then there are other changes, like the accumulation of abnormal material. Those are called storage disease. I think I'll show an example of that. There's a long list in several animals, but we'll see it later. Then we can have selective loss of neurons, or even, you know, like altered neurons or axons due to neurodegeneration. And also you can have spongy states, which is the feature observed in case of edema, spongy degeneration, or like spongiform degeneration, like in prion diseases. But sometimes they can only be observed as artifacts. So if the brain is very autolytic, if it spends too long outside the formalin, sometimes you can see these holes in the brain, which of course don't have any significance. And in other cases, you can just observe it in older animal, where like it's a incidental finding. Usually they don't have a meaning in terms of diagnosing diseases. But it's important to know that they are actually artifacts. It's very important to know artifacts because they don't have to be confused with what the real lesions are. Those are not the only tool we use in diagnosis. Of course, you have to be knowledgeable in several to observe several other patterns of distribution, for instance. So the localization of the lesions is really key. So what I was saying at the beginning is that, for instance, in a liver, like a lesion can be, and usually is spread out. So you can see it almost everywhere in the central nervous system, but in the nervous system in general, it is of paramount importance to understand the localization of the lesions. Unfortunately, in farm animal, there's not many clinicians, or we don't receive as diagnosticians like clear indication of where the lesion can be localized. But for companion animal pathologists, sometimes it's easier. So they receive already like an indication where they could go and look for the lesion, which is very helpful. So the localization, the distribution, so is the lesion spread out everywhere, is generalized, is localized in the gray matter, is localized in the white matter, is localized in both. Is it unilateral, bilateral, symmetrical, non-symmetrical? All these features are very important to reach a diagnosis. And then there are certainly other information which are very important for any diagnostic pathologist, which are the species, the age, the breed, and also the clinical history that include vaccination or like a previous story of a similar condition that you're observing in the case you receive. So all that are very important aspects for reaching a diagnosis. So, well, basically, how important is the localization of the lesion? Well, it's as important as for you everywhere in the world you're now to know that the place where I'm speaking now is basically in Europe first, then like going down to a smaller area is Great Britain. But then is Scotland, and within Scotland is like we are now in Midlothian actually, but here I had a picture of Edinburgh just to give you an idea, like of more than anything of how you step by step you get closer to the place where you want to go. Similarly, in the nervous system, you have to know where you're gonna find the lesion. Are you gonna find the lesion in the brain? Are you gonna find the lesion in the spinal cord, in the peripheral nerves, or like even sometimes in the like ganglion? And to be honest, in many cases, even examining the muscle, even though of course it's another organ, can be helpful to finalize a diagnosis. So that's a very crucial, important part to know where you have, you may expect your lesion. Now, as I said before, most of the time we don't get so lucky. We have phenomenal colleagues. We receive cases from the SIUC service. This is the veterinary service in Scotland. They're great colleagues, and if they have the chance to examine the animal, they can give us an idea if the condition they're observing can originate from the brain, or if they can be maybe originating more from the spinal cord, or they may not be sure. But sometimes, and this is quite a large percentage of cases we receive, the animal is just found dead or dies sadly, which suddenly may mean that the animal was observed alive a few hours before or even the day before, healthy and normal, and then the day after was found dead. So we really need to examine many parts of the brain to be sure we get, like we can see the lesion if present. Like it's not that we get lesions in any case we receive. Actually, well, I'd be delighted if it was so, but many of our cases are just negative cases. Well, though I want to stress that a negative case is also a very important case. Why? Because there are diseases that you want to rule in or out. And so it is sometimes a negative result is almost as good as a positive one. So the way we do it in our lab each lab may have a different strategy. So basically, you know, some lab may have slightly different sectioning of the brain, but overall, I'd say that for ruminants, the area you see here are the area that are sample, like they are like pretty good to have to reach as close as possible to the diagnosis. So basically, like what you'll see here is there's like those lights in this picture. So the histologist, or maybe that was me, I'm not sure, but cut like decently thin slide, and then they put this light, well, and then we put, you know, like we had to section them a little bit and make them slightly smaller to fit them in this plastic cassette. So once you put them in the cassette, they're gonna be processed and are not gonna enter into like too like fine detail. If there is an interest, I have some other presentation where I have more detail over processing of the tissue. But in this case, just, you know, just to give you an idea how all the process works. So this brain is put in this cassette, and eventually you get a block with paraffin wax to keep it, you know, to be able to cut it. And then this very, very thin slice are put in a slide, and so they're gonna be ready to be looked under the microscope. So as I said, the localization of the lesion. So as you know, you've just seen that we cut those slides for each area. You can see here roughly the area we section, and well, I'm very grateful to Welker Wisconsin Collection Atlas, where I could grab some picture of the area of brain that we use in our lab and most labs. And again, as you know, like very helpful to get a bit of orientation where you are in the brain and how it looks like, to know, you know, like where to find the lesion, and once you find it, to know which kind of disease cause lesion in that area. So now just we'll start to have a quick look to, you know, like we'll just see the relatively little you can do with the brain in terms of macroscopic examination. Why is that? The reason for that is usually is very important to have the whole brain fixed. If you start, you know, like sectioning it when it's too fresh, that is gonna alter, distort the appearance and the anatomy of the brain, and then making things pretty difficult. Now, in certain circumstances, we have done it in the past. Like literally, when we were doing this study, we would have split the brain into two halves, one for the histology and one for the molecular study. But most of the time, if it's just a field case, you grab the whole brain, collect it as well as possible, and put in a decent amount of formalin for many days, depending on the size of the sample. But sometimes the altered, the pathology is so clear that you can see it even almost before fixing. So just, you know, to show you what I mean, and that's a picture taken from a paper from some very dear colleagues in Leon in Spain, and that's about a condition which is, as you see, like of a Chura lambs, which is a breed typical from the area where the publication was put together. And so I just wanted to show you that and how important is to have a comparative example, if not in front of you, in your mind. So this is the appearance of a normal matched age lamb in here. And just next to it, you have the altered brain, where, as you can observe, the gyres of the brain are not so visible. So the cortex is almost more flat. And that is like called lissencephaly. There are several degrees. I think I have another picture with a bit more extreme flatness of the brain. And also in this case, and I think you know it can be quite easily appreciated, there is hypoplasia of the cerebellum. So like I found it, this comparative picture, like brain one to each other, very important, because to understand pathology and changes and lesions, you need to see and to have a good idea of normality. Of course, normality have a range, you know, like there is not the ideal normal brain, but when you've seen a lot of brain in lambs of different age or bovine or any other animal, you are familiar to what a non-altered brain would look like. And this is your starting point to then go and think and see that something may go wrong in other brains. So as I said, you know, like some example of a bit more extreme, possibly malformation. So like kind of similar, but like in this case, you have like a tiny cerebellum, well, a small cerebellum, the part of the almost brain is almost exposed, the brain stem, you shouldn't almost see the brain stem, even though this is very altered, should somehow look a bit more like that, but there is a clear hypoplasia of the cerebrum, the hemisphere in this case. This case, it's even like a further like lissencephaly, quite extreme, almost is virtually impossible to see like the structure of the gyres. And in the third case, this is one of our cases, is like an extreme form of hydrocephalus. The hydrocephalus is so severe that the brain is basically almost like a very, very thin paper almost, because it's full of like liquid, basically. So when you try to extract it from the skull, it almost breaks down. It is challenging to fix it properly because of, as you can see, of the structure of the brain. But still, it is interesting to examine it, and if possible, you know, also under the microscope, even though you already can see macroscopically that this is, you know, like a malformation, but sometimes you can get better hint looking down the microscope. So about malformation, there are several different causes for malformation. Some of them are insult during pregnancy. So in the uterus of mothers or ewes or like cows. But today, just for the time availability I have, I wanted to concentrate just to show you a case story about what viral infection and teratogenic viral infections can do to the brain. So like, as you can see in this table, basically there are just examples. Those are not the only teratogenic viral infection. I concentrated on the one that are somehow similar or dissimilar, and some that I had basically the experience to and to diagnose, you know, personally. So, you know, like one of the virus that cause nervous system changes and actually central nervous system changes is the Acaban virus, orthobunia virus. The blue tongue orbivirus can also cause altered anatomy, while malformation have a teratogenic effect in the central nervous system, and the pestivirus, BVD and BDV, which is possibly like those are the cases I've seen more in Scotland since I'm here. So what do they have in common and not in common? So like in terms of hydranencephaly, which is almost what we've seen in the previous slide, where the brain was almost destroyed and flattened and open. This is seen in orthobunyavirus and in cases of BTV 8 usually, because this is the strain that gives central nervous system alteration. But for instance, in case of pestivirus, this is not a feature we'd observe very commonly. Whereas in terms of the cerebellum, there is this genesis, which is more intense in case of pestivirus. And then, of course, there are other indications when, like when the animals are born, that there may be something like quite wrong going on. We'll see some picture about that. And then there are other fetal congenital presentation, depending on the virus. Like, for instance, blue tongue, you may just only observe abortion. Like in pestivirus, you observe abortion, but also other aspects of the disease, like weakness at birth and nervous signs, and all that is associated with hypomyelinogenesis. Whereas in the orthobunyavirus, you have weakness at birth, and then the weakness you see at birth is associated more with polymyeloncephalitis. So, like, the reason for showing that is just to tell you that, when the clinician, when the vet or the farmer observes some condition, some altered pattern, you may, have similar presentation, sometimes, just abortion, and then you have to make a story and basically get to a diagnosis. And that, as I said, is a quite nice story, like, well, it's not very recent now, but well, in terms of science, is decently recent. So like 9 years ago, a new condition emerged. It was observed first in Germany. My colleague there, the first thing that they observed was a drop in milk production in dairy cows, and they basically also showed some fever, and that was it was a bit worrying, and not well, they didn't get to an immediate diagnosis, but I must say that they have to me really commented, because very quickly they basically isolated the virus and sequenced that, and that was a very successful, very big success of how the diagnosis and collaboration between diagnostician can work. So in the same time, though, after Germany, in Germany they observed these cases. In Holland, they started to observe similar changes, but they included also diarrhea and other feature in the herds, but also they started observing malformation in calves. And I'm very grateful to Klaas Pepperkamp and Willem Huda for sending me this picture from their lab, which I had the privilege to visit last year. A fantastic place. They do especially research and diagnosis in farm animal. So I think it's you can see that just very easily that there's something really, really wrong with this lamb and this calf. They have arthrogryposis, both of them. And then when the animal were examined, and so the post-mortem was carried out, they were found to have hydranencephaly, microencephaly. Some had cerebellar lesions, some others haven't, and also poliomyelitis or myelodysgenesis. So like quite striking lesions. And basically those disease and virus was called Schmalberg from the place where it was first diagnosed and sequenced. And then it started spreading out. Here, just another couple of images, still from clamps, and can show you, like just to see. So that's the skull, the ear, like of a bovine and the lamb. So, like, I think you can see there's almost a hole inside the brain, a lot of space. The cerebellum is tiny. This is a histological image of the cerebellum. Unfortunately, I should have had a normal cerebellum next to it for the same reason I was explaining, that you need to know how it looks normally. But trust me on that, there's like a lot of alter pattern. This is like not what cerebellum should look like. And even more so, the spinal cord, basically, you should be able to see the dorsal and ventral horn, but here the ventral horn are almost non-existent. And this is a feature and the pattern of Schmalberg disease. So, well, I'm not gonna spend much longer on Schmalberg, just to say that thanks to my colleague and to like the good communication there was, I was able to diagnose the first case of Schmalberg in Scotland in collaboration with our virologist here. So it is really communication is of paramount importance, like communication and connection between colleagues all around the world, because you never know where something is gonna come from. Now, setting aside for a moment like viral condition, going back to the macroscopic observation you can do to diagnose certain disease, like there is another one which is the Echinococcus granulosus. As you know, like as you see, it is quite striking. So usually we don't receive cases of Echinococcus. At the beginning, when I arrived in Scotland at first, I thought, do you have the disease? Sometimes, in certain countries are free of certain diseases, but then I was told that what happened is that the practitioner, so the vets, would have seen them on their own. They wouldn't need to send it to me. Fortunately, sometimes, in case of acute, initial, very preliminary neurosis, I was submitted some brain, and so I had, I had a chance to observe it under the microscope. But this is a quite rare event. Well, that's a parasite, Tania, and has quite a complex life cycle that involve dogs and sheep, and also can end up in human. Certain type of Echinococcus can end up in humans and cause, like quite severe lesions. Now we move to space occupying mass. As I said before, tumors are like feature that can be change that can be observed macroscopically, but to be honest, in ruminants, tumors are not, well, fortunately, they're not very common. They're much more common in human pathology or companion animal, pets. This doesn't mean that farm animals don't have tumors, but it is a much more rare event. The more common finding are abscesses. I think I'll show you some, or granuloma. and they are, again, very easy to be spotted macroscopically. So you see this is like an enormous abscess caused usually by bacteria. And again, most of the time we don't receive this kind of case because it's so clear to the clinician that, you know, like all they do possibly is maybe to take a swab of the pus present there and maybe send to the lab and see if they can find which bacteria is involved, maybe to treat the rest of the farm if it affects more than one animal. But sometimes we do get a case similar to that. So this is one of that. So you see there's a large abscess. It was not as easy, sorry, I should have shown you the whole brain before. So it was not so obvious as it was in the previous case. So once we fixed the brain, we could slice it. This is a case from back to 2016. And we found like really, really large abscess. So like macroscopically, you'd already see them. But microscopically, down the microscope, I could see like other small abscess in other areas. Again, I didn't have the chance to observe this kind of lesion, which is like a granuloma. Now, just by the looking at it is very difficult to see. Sometimes you can figure out what you're looking at by the color of the mass. So usually it's more yellow. If it's just passed by bacteria, can be a bit more greener. If maybe sometimes parasites are involved or fungal condition. And in this case, well, I know, even though this is not my case, but it's a case from my colleague, and I know that this was a case of fungal infection. I'm not sure if it is easy for you to see that. So this is fungal, so this is a knife. I think that was an Aspergillus, if I recall well. And it's a shame I don't have a broad pot. Usually we use like a special staining for spotting even better in the tissue, like the agent. But in this case, they're quite, you know, evident. So just to show you that, you know, you can see them macroscopically. Again, I said, you know, like in case of myelin disorder, when there's a myelin disorder, you can see powder and sort of consistency of the white matter. And one of the disease I got to see quite a lot in my career here at Moredun is called Swayback. And this disease is usually due to copper deficiency. So there is a deficiency on the supply. It affects more young animal, young lamb and goat are affected, but to be honest, I've seen it in, you know, like more adult animal. And what, you know, like how this disease work, copper, like interfere with the formation of white matter, so of the myelin. And like, because, you know, like the myelin is not formed, this gives symptoms, which are, you know, like quite clear. The animal, you know, may die. And so when we receive the case, we sometimes are lucky enough to see macroscopic changes and microscopic changes, but many times I only see microscopic changes. Now again, this is a really striking picture I was given from a colleague. Like quite some time ago. I think it's really like illustrative of how even Swayback can be, you know what we were saying before, where the lesion is localized. So localized is unilateral, bilateral, and there are some conditions that they can show like in different ways. So like, for instance, in this case, Swayback, you see in this series of section, is more unilateral. So you see this wee hole, like small holes here. So it's like unilateral, whereas in this other brain, it's like very severe. The myelin is almost virtually disappeared, leaving holes, and it's bilateral. Microscopically, what you see is a rarefaction or disruption of myelin. But in milder cases, so and also in more like not super young animal, what you see is like chromatolysis, chromatolytic neurons in the red nucleus. This is basically pathognomonic. So when I see this lesion, I'm pretty confident that's due to Swayback. And now we go to examining Malaysian necrosis. I think I have just some example. I don't have any, I think, Tromor-Emfert example, but I have an example of another quite common disease that, again, I diagnose quite frequently, unfortunately, which is the focal symmetrical encephalomalacia. It takes its name from the nature of lesions that are apparent in the brain. It's caused by a toxin by a bacteria, Clostridium perfringens bacteria, and it basically altered the vascular component in the brain, and basically what, so just in this image, if you see like the red spot is to show that it's, its distribution in the brain, which is, like is either in the frontal, you can see it in the internal capsule, or even a bit more in the hidden brain, like brain stem. And I thought that was a very good example of what this disease can do. So you see here, there's almost, there are no holes, but you see this large spot. Those spots where the tissue is really soft, there are almost, say, Emfert, and that's, what those lesions, which are symmetrical, focal, they're symmetrical, encephalomalacia, and this, as I said, malacia, so necrosis, which down the microscope, it's usually seen like that. So you see this paler like area. It's because the tissue and lots of cells are basically dead, and there's also hemorrhages, like small hemorrhages. But another clear feature that sometimes is the only one I observe is this intramural serum leakage. You see this like really pink, glassy serum. This is, again, like, depending on the distribution, the quantity, etc., is decently pathognomonic of FSC. As I said before, it affects more lambs and kids, goats, but I've seen the condition in bovine too. Another condition, sorry, I need to the water. So another like metabolic condition that you can observe is the what is called cerebrocortical necrosis, CCN, which is also known, and actually is more popular, like polyencephalomalacia, especially in US. And the reason for that, again, it is a name from the distribution and type of lesion you see in the brain. So it affects the like gray matter, polio, which is like the gray matter, and leuco is the white matter. Encephalomalacia, because it causes necrosis, but this time, instead of doing it mainly in the white matter, it does it in the gray matter. Also affects like sheep, but this condition is actually observed in cattle, goat, and deer too. The young animals are more subject to get the disease, and this condition is caused by altered thiamine B1 B1 alteration or sulfur toxicosis. It is pretty difficult. Some colleagues believes and say that there is a clear feature to distinguish between the deficiency of thiamine or the toxicosis. I really struggle with that. I find it difficult, and most of the time I think that the history and other findings are more helpful to establish which give origin to this altered feature. Now, the curious, histologically, what you'll see differently from the previous one, the previous one we saw it was the core, so it was this part, the white part that was necrotic. In this case, is the cortex. So that's the cortex, and I hope you can appreciate the difference. This side is more, say, normal, or from this magnification you wouldn't spot an altered area. But in this other side, where you see a paler, almost line in the brain, that's the area where you have the necrosis, which is usually called laminar, pseudolaminar necrosis. And if you go down a bit in the microscope, higher magnification, you have neurons, those are neurons which are almost shrunken and triangular. Their cytoplasm is not the most easy thing maybe to see in this picture, but it's more pinkish. It becomes more eosinophilic because basically the cells are dead. And really quite curious, I would almost call it trick, but well, it's not. But another diagnostic tool you can use is once you slide when you cut the slide in the brain, you can expose the brain to an UV lamp, and the area that are necrotic are fluorescent, and so you see here, all that area here would look down the microscope like that. Now, I don't know if it's totally clear to everyone, but I just want to highlight that this is exactly the identical slide. So this brain is the identical brain. The only difference is that before taking the picture, in this case, it was the UV lamp was turned on, and if you see, they are identical, and that's where you could spot the changes. Sometimes the clinicians, the colleague, are happy enough with this feature to have a diagnosis, because usually when you see this kind of fluorescence, it's almost diagnostic. But the point is that sometimes it's not so clear, sometimes it's very localized, and to be honest, sometimes it's not present too. So fortunately, we receive the brain for histological examination. And now we go to the microscopic examination. Of course, with the macroscopic examination, I could be a bit more exhaustive. I mean, examine in many cases. In this case, I have to be really, really short and focused because there's so many diseases that, it would be virtually impossible to explain all of them today in this event. But I just want to, highlight the more common and interesting one. So, first of all, well, maybe just before going deep into the just histology one, there is a condition which is halfway. Similarly, what I was saying before, there are conditions that you can see macroscopically and then microscopically. This is the case for suppurative meningoencephalitis. That's, if you see here, there is pus on top of this area, which is more yellowish. It's very common, it's much more common in newborn and young animal, and it can be caused by a list. This maybe is not the most exhaustive list, but those are the most common bacteria that can cause the disease. And most of the time, we hardly can distinguish between these bacteria microscopically. So basically, colleagues in the bacteriology lab, they do testing to establish which bacteria is involved. But sometimes the bacteria have a very specific and precise feature, like is the case for the Staphylosomnus. And because this bacteria cause a very different feature pathologically, and as you see, like it causes like very severe actually feature. Like in this case, you see like this, you have like quite a severe perivascular cast, but also hemorrhages, and all around there is like necrotic tissues. And what this disease causes is the formation of thrombus. So the thrombi then cause infarction in the brain, and that's why you have this quite, like specific feature down the microscope. Haemophilus is like a small rod-shaped gram-negative coccobacilli, and this is mainly seen in cattle, but sometimes is rarely observed in sheep too. And then when I was, preparing this presentation, I thought would have been quite curious to see like different cases, like different example of condition we receive in this lab here, with an exception, because one of these diseases is not commonly present in this country. So like again, a Spanish dear friend of mine provided me with the slide, Julio Benavides in Leon gave me some slides from his PhD study thesis. However, I guess that, like just because the difficulty of doing diagnosis in brains, even, like I said, like diagnostic pathologists in other organs, sometimes they feel a bit itchy in front of like neuropathology. And I must say that sometimes can be, at first confusing. So if you see those cases, it's pretty difficult at first look to establish if there's almost virtually any clear difference between them. But I can tell you they're very different diseases. So what are those? So we'll start from the first one, the A1 on top. This is a condition I find myself to diagnose quite a lot. I must say I like it a lot. Well, not for the animal, because this is very unpleasant, and actually is a zoonosis, so it can affect humans too. But for me, it's a very rewarding disease, because once you're familiar with the condition, and if it's a form which is like decently typical, I can, like have a diagnosis very, very quickly compared to other conditions. And basically, this is the disease is listeriosis. Listeriosis is caused by a bacteria, a gram-positive bacteria. In this country, there is a quite high incidence during the winter, so like in Scotland, in the UK in general. And this caused by like low quality silage. When the pH is not ideal, sometimes there is an overgrowth of listeria. Listeria is basically like everywhere. It's not a commensal quite, but almost. So less there is a bit overgrowth, and then if there is, the animal can get infected. And even more so, it happens in coincidence with permanent teeth eruption, because so there are like minor lesions in the mouth. The bacteria can just like step in, enter through the nerves, and usually through the trigeminal nerve, and get to the brain stem, where it causes the disease. So as I say, because we know that's the case, it is very important to see where we observe the change. And usually when we have cases of listeria, the majority of the more severe cases are around this area. Occasionally, I've seen some lesion also in the more frontal brain, And I started seeing that like one curious feature, which is not like clearly maybe like so much described, some like mild vacuolation in the internal capsule, but the core lesions are in this area. And just to show you, battery is running low. So so said that, as I said, is very important. And what do we see, and what do I see under the microscope? One of the like more characteristic, I would say, in certain form, in subacute, acute, subacute listeriosis, are micro abscesses. If you recall, I spoke about abscesses at the beginning. You have this large, like almost like, you know, enormous macroscopically visible abscess, and then you have the micro abscesses, which is like, this is the case. They are smaller. They are composed mainly by neutrophils, which are, you know, pyogens cells, but also you have like thick perivascular calves. I should have put maybe an image with a close-up. So like the vessel is surrounded by mononuclear cells. When the listeriosis is a bit more chronic, so later on in the disease, sometimes you may not have this nice micro abscesses. So it can be slightly more trickier to say like 100%, to be totally sure that it's a case of listeriosis. Most of the time it's quite a nice, like, well, decently straightforward disease. However, it's pretty good to have further tool to diagnose and to refine the diagnosis. So in the lab, the first thing you can do, like the easy one, is a gram. If you're lucky, you have still quite a decent amount of listeria, as in this case, you see like the blue dot, so the red dot are the nuclei of the cell. The blue dot, this like small rod here, are the listeria, but sometimes there's really a sparse presence and scant amount of bacteria. So it's very handy for us to have an antibody that works against listeriosis. So with the immunohistochemistry, with this technique, we can basically like refine and have a more precise diagnosis. We are working now with some colleagues to use this immunohistochemistry that we usually use in the brain, also to apply in other organs, even the gut, but we are still at the preliminary stage at the moment. Now we go to condition B. So in this condition, you won't see like the micro abscesses. There's no suppurative component. This is a mononuclear condition. However, as I said, in very chronic listeria, you can have like similar calf than that. So what's different in this disease? The difference is already here. There's almost all the components that allow me to diagnose this condition in this country. Even more so, this is even more important to say, because this disease is virtually almost just present in the UK, actually in Great Britain and like Scotland and Wales. And what is apparent down the microscope are this like perivascular calves, like quite lymphocytic-like. Then you have dead neurons, so neuronecrosis, and then dead neurons that are eaten by macrophages, and it's so neuronophagia. All these feature together. Sometimes you have glial foci. Here is not totally clear. Like maybe this one, where you see like microglia forming, you know, like in groups, and that's, you know, like quite a typical feature of what was saying Laupinell. Laupinell is an acute encephalomyelitis of sheep. even though doesn't affect just sheep, but it does affect mainly sheep. It is caused by virus transmitted by ticks, and it is part of the complex of the tick-borne encephalitis. As I said, it can affect other animals, can affect cattle, deer, alpaca, pig. It's been described in many other animals. Recently, there was, I think, here diagnosed in the viral surveillance unit in dogs, but it's also a zoonosis. Fortunately, so far, there haven't been many cases in humans, differently from other tick-borne encephalitis. Excuse me. And several cases of the condition was almost caused by handling the virus to prepare the vaccine for the condition, so lab iatrogenic condition in man. As I said, this is like mainly a UK disease, but it has been very occasionally, sporadically reported in Bulgaria, Turkey, Norway, and Spain. The localization of the lesion, again, very similar to Listeria. Again, the brainstem, hindbrain, and sometimes the cerebellum, even more so in the laupinale, are affected. Similar areas, but the histology features are different. Here, I think we can see it even better. Dead neuron necrosis, maybe a case of starting neuronophagia and glial foci. Possibly the chance are that a neuron was here before, but now have been taking over the phagocytes. Tiny perivascular cuff, they can be larger, we've seen before. And here the neuronophagia, an example, a very, very clear example of neuronophagia with this like large cell neuron dead, which is being like eaten, like get rid of by macrophages, well, phages. Likewise for even though usually it has like quite pathognomonic features, it is good to have further tool to confirm the disease. In the cerebellum, you can also see like death of Purkinje cells, sometimes infiltration of inflammatory cells. But it is pretty good to have an extra tool, which is the immunohistochemistry, like here. This very, very dark spot, this is the virus inside cells, and inside either axon or dendrites, those are dendrites of the cells, which help to confirm the diagnosis, which is a very helpful tool in diagnosis, especially in cases like this one. This case has a special story. And it's a case I got back in, I think 2010. I received the case, and when I looked, because I received just part of the brain, so as I said before, usually it's important to have the whole brain. You cannot really make a clear diagnostic by just a part of the brain, but at the beginning I only received part of the brain. And in the brain, I observed I couldn't really clearly see any proper infiltrative inflammatory change. And the more striking changes were this swollen axon in this area of the brain. And I thought that's a bit strange. Maybe it's toxic. And then I just sent my report saying I couldn't reach a clear, a more refined diagnosis. So my colleague that sent the case got back to me and told me that the animal had really, really very high immunoglobulin against laupinale. So the serum was tested. It was really positive. And if there was any way I could double check if the animal could have had laupinale. So I asked for immunohistochemistry, being a bit in doubt about the results. but the results were absolutely amazing. So like it was covered, it was like just like full of like virus. And actually this case is now one of our best positive control. Possibly the reason why I didn't see any lesion is because it was a very acute case. So just when the virus invade the brain, and so the quantity of virus is very large, but then after some time, you know, there may be a reduction once the inflammatory cells take over that. Next one, the C, that's the one, as I said, it's courtesy of University Leon, the Department of Animal Health. And that's slightly different. The thick cuffs are not, you know, like are not in the middle of kind of normalish brain. So there is like almost a pale appearance of the tissue around the cuff. The reason for that is because this virus, which is Medivisna in brain, cause altered the myelin, give alteration of the myelin. That's a chronic disease, usually of adult sheep, and it's a retrovirus, lentivirus, and it affects more than one system. So it can affect the mammary gland, the lungs, the joints, or the central nervous system. The nervous form is not hugely common. It's been observed like quite a lot in Spain in younger animals, roughly around 2 years, because, you know, like it's a lentivirus, it takes some time to form lesions. It has been, though, described in other areas around the world, not very many, but it is important, you know, like to keep an eye on every possible condition you can observe to have your differential diagnosis. And it has a really interesting feature. Usually it can be monolateral with this like almost triangular lesion with cuffs and altered myelin. So here we have a macroscopic picture of the cerebellum, and you see here there's some like strange altered tissue, like almost, and I can tell you like in real life it will be a bit soft. And what you see down the microscope is like you have basically malacia, necrosis of this area. The myelin is affected and forms these large, severe lesions. Microscopically, again, you see the cuffs, slightly different than before, affects the myelin. And again, like there is a tool which unfortunately we don't have in our lab any longer. We used to have this antibody. It has been, the production has been dismissed, so now we won't be able we won't be in the position to be able to diagnose it. But it's, you know, that's a picture that was sent by Leon. They may still have some antibody left. And now we get to the last condition, which is possibly within those the most curious one. This form of this disease is very rare, but as you see, very severe. And there is feature of a bit all of the other condition, with the exception of like possibly listeria. There's no suppurative component here either. Sometimes you can have maybe some eosinophils, but it's not very common. You have more, again, a mononuclear meningoencephalitis or even encephalitis with cuff and glial foci. This is a case of sarcocystosis. I can tell you that was sarcocystosis. It is curious because usually sarcosis doesn't cause like severe lesion. This protozoa just like go to the tissue, even to the brain, other tissue. The heart sometimes is like really full of even large cysts, and it literally doesn't, you know, interfere with the life of the animal, doesn't cause any lesion at all around it. But in certain circumstances, which is not 100% clear to me yet, it can, you know, the cyst can break and then can start causing inflammation up to, you know, like the point of reaching the case we saw in the first image. Usually the reports in the case I've seen here affect more the spinal cord than the brain, but the brain can be also affected. So now, just to summarize and to show you how important it is to know where the localization of the lesion is and the specific characteristic that distinguish one from the other disease, like micro abscesses in this case are really very nice to have a good quick diagnosis. Neuronecrosis and neuronophagia is another one for Laupinel. Like the distribution and character of lesions, and in case of sarcosis, we are a bit less fortunate because there's no immunohistochemistry or special staining. In this case, we also have to hope to get lucky and find the agent, the parasite, but it's not always so easy. And we've been working quite a lot. We had a PhD student, Tanya Lepore, that tried to produce an antibody for that, but unfortunately we were not successful. So after this flavor of how complex and or even if looked with a systematic way certain disease can be, I'll just mention some very important conditions that cause changes. In this case, mainly in my experience, I observed them in the fetal ovine in case of abortion. So I should have maybe said at the beginning that at Moredun, our diagnostic service is focused more in neuropathology and abortion condition. So we receive cases of aborted fetuses, sometimes placentas, and the lesion we observed in case of toxoplasmosis and other protozoa with the definitive host cards that cause abortion in ovine, but also not just abortion, clear lesion in the central nervous system. So those are the characteristic lesion, glial foci with central necrosis, in this case even mineralization. In other case, you only see leukomalacia, so a necrosis of the white matter, a lot of congestion in this case, and depends on the stage and the disease. You can see a slightly different feature. In this case, I haven't shown, but we were lucky enough to develop an extra tool. I forgot to put an image, but we now have a good antibody, antiserum. It's like a polyclonal for very specific for toxoplasma, and we have also another one for neosporosis. Similar disease, protozoal disease, affects more cattle, affects cattle and cause abortion. It has a different pathogenesis. The definitive host is the dog, but the lesions, as you can see, are similar. Well, okay, there's no mineralization in here, but glial foci with internal necrosis. And this is an example of how cyst can label and how cyst can be spotted. To be perfectly honest, I think this is the brain of a dog, kindly donated by Professor Carlo Cantile from the vet school in Pisa. But the reason for choosing it is because it's, the positive is much more clear. If it was this case, for instance, what would I have expected is to see positivity in the central part of the glial foci, but would have been much more difficult to show you. And last, just a slide of an extremely important disease, which is rabies. I don't have much experience. Actually, I should say I don't have any experience of rabies because the UK is free of the disease, so there's no cases here, but I thought it was very important just to show you actually in you know, other countries is present, and it's a like dangerous disease, it affects humans, and it causes the formation of negri bodies inside neurons, in the cytoplasm of neurons, in different areas. So like, just I want to mention that in carnivores, which are usually the mean of transmission to humans, like dogs or cats, not so frequently foxes, this negri body localize in the neurons in the hippocampus, and they change their like behavior. So that's why, you know, like there's the form of the furious rabies. The animals start biting more. They're scared about water. But in ruminants, the lesion in the negri's body is localized in the cerebellum. So usually it causes a different pattern of clinical disease, and it's more like a paralytic pattern. So the animal are more like ataxic and more stealth. And it's the place, so it is important to know, because it's, you know, it's the cerebellum, the place to go and look for this body, inclusion body, this formation. We move now to the storage disease. I told you I would have explained you like storage disease. There's a long list in several animals. You know, like I'm not going to enter in any detail of that, but our disease I'm very interested at, and I've been wanting to diagnose one for a long time, and eventually some years back, I cannot recall exactly when, but a dear colleague of mine, Helen Carty from SIC, sent me a case from a Jacob sheep. So those are this like really beautiful breed. Usually they're kept as hobby flock, and I started picking like a strange feature in the neurons, like even like either large neurons, but also in the cerebellum. And that's so, excuse me, like a strange like substance inside. Like, so basically this is, you know, accumulation of storage disease, is an accumulation of like different material depending on the missing enzyme that is like due to basically digest this condition. There are also like condition in humans. Those diseases are studied quite in detail for, you know, like for kids. And actually, well, the reason for me for mentioning is because we wanted to study this condition a bit more in depth, but it was very difficult to find someone that would have examined our material. And we've been very lucky that Professor Tim Cox from Cambridge University accepted to carry out a collaboration with us, and they've done a restriction enzyme analysis in Jacob sheep tissue, and they established that it was like, basically this is a genetic condition, transmissible. And the good thing is that through this collaboration we were able to develop a tool to test male animals to see if they harbor the condition, and so to rule out them from breeding, and so basically somehow getting rid of the disease. So just you know, like I thought it was a quick, nice story to show how, like, from diagnosing a disease you can up, you know, like maybe solving a problem. And finally, we'll just have a quick look at the spongy state changes. So as I said at the beginning, that's, you know, like cause edema. So like almost like enlargement, content of fluid inside the brain. So expansion, spongy degeneration or spongiform degeneration. And the, as I said before, artifact or old age. I'm not going to show any artifact in this case. Just to introduce you very briefly about the TSE, so the transmissible spongiform encephalopathies. Why are they called like that? Well, the name say it almost all. these diseases are transmissible. They are like transmissible within species, but also between species. I've been engaged on the study of those diseases for many years. We used to have like a group studying that. I was leading the group up till when we stopped doing the research in 2012. So that's a picture from back then. So as I said, they are transmissible and they're called Spongiform because of the vascular aspects that neurons have down the microscope. So like they resemble a sponge because of the hole. And then there are encephalopathies because the main organ affected is the central nervous system. The prion can be spotted, so the altered protein can be spotted in other organs, but the affected organ with lesion and symptoms is the central nervous system. One of the most well-known, common, especially like in the UK, is scrapie. That's just like scrapie is called like that. Like this disease cause the animal to be really very itchy and scarred to scrape, like to scratch themselves. You see here they're like hole missing because it scratched so badly that it caused the hole to be almost get rid of in this area. But then the animal also lose condition and start being ataxic, depending, how severe the condition is developed. And here you have like an immunohistochemistry of PRP, the prion protein, altered prion protein, and like the spongiform appearance of neurons, but also white matter in the brain. And just like mention that there is, so this is the classical scrapie. There is also an atypical scrapie, and again in my lab we diagnose the first case in Scotland of atypical scrapie. This is slightly different, is observed more in the cerebellum. So the cerebellum, that's the molecular layer, and you can see all these tiny holes. And again, when we tested it with the immunohistochemistry, we found it very positive. And then another disease that fortunately I haven't diagnosed and is still not in the UK is a condition known for a long time in captive mule, but this is quite a tricky but disease because within all the TSE it's very contagious. So the animal can transmit the disease to each other through saliva and urine, which is not the case for BSE, fortunately, because it's a zoonosis, and not so much from scrapie, where the transmission is more through placentas. So finally, I get to the bovine spongiform encephalopathy. Most of you that are here must be like a bit too young to remember the big outbreak of BSE that caused that's a case, this poor animal that's were losing condition and getting really quite bad neurological signs. Entire hairs had to be get rid of. An enormous amount of animal had to be killed and get rid of. And as you maybe be aware, this is a zoonosis. So through the consumption of meat of those animals, certain people can get affected, and this causes a disease called the variant Jacob disease, which is a disease that develops along a long time, but there's no cure, and it just kill with very severe neurological unpleasant symptoms. And just to conclude my presentation, at the time when I was preparing this presentation, I just would digit veterinary neuropathology, to do some search of some picture to be, you know, like a bit more as illustrative as possible. And it was, you know, with like great pleasure that within the very first image that appeared, well, a part one of the main book, which I had with me, this is, you know, like even though now it's not super new, but this is, say, our Bible for neuropathologists. There are also pictures of two, like, very dear colleagues of mine and close friends, who happen to be my supervisors in my career. So, like Martí Pumarola from the vet school at the UAV University, like neuropathologist. And he's the, well, now colleague, then supervisor that I carried out my study on lymphoma on central nervous system of cats. And Professor Carlo Cantile, who is from the vet school. I've done my thesis dissertation to become a vet. In Italy, we do a thesis. And Carlo is also like an amazing like neuropathologist. He's the author of the, is one of the co-author of the part of neuropathology for the Jobson Kennedy book. And again, like every pathologist, not just neuropathologist, there's 3 volumes of that. It's quite a substantial book. So it was really a pleasure to see like they were somehow like really listed like between the first search. So it was an enormous privilege for me to be trained by them, and so like I want to take this occasion to thank them, and of course to thank you all for listening to me. Hopefully, you know, everything went as well as possible. As I said, if not, the presentation will be recorded and you'll be able to like see it later on. And I'll also, you know, like look at the question and see if there's any question for me to address. So I just like just thank you all again, and hopefully I'll see you again soon.
All right. I'll just follow you.
Oh, sorry. Okay, so sorry, I just like, well, one of the like tricky thing to be like live online. So I'll just check his, I guess I can see.
There's quite a few people who that's problems with getting involved, so interesting here. So any questions that you like? So,
okay, I'll have a look at some question to see if.
This one here. I'll keep them up on as well. So if you just.
Okay.
See one? Yeah. Translations.
Okay, so I have a question asking what are the major parts for studying neuropathology ruminants. I'm not highly sure I understand it in which respect. I guess I'm not sure if it's which is the most important part of for the neuropathology in ruminants. So I would say, I'm sorry if I'm not addressing the question properly, but differently from, say, like pet or like companion animal, like the diagnosis in farm animal is slightly different. So basically, usually what is very important is to diagnose in numbers. What I mean is that, sorry about that. So in farm animals, what is really important is to have a quick specific diagnosis to allow the farmer and the vet to deal with the potential damaging outbreak in the farm. So usually the individual itself is not hugely important, as it is, for instance, in, of course, human pathology or small animal pathology. If you have one, say, one-off case, this is not gonna affect your herd or flock very much. So if, say, I diagnose a tumor, that is one-off. Maybe interesting, I may even get to publish it, but this is not a condition that is of major concern to the farmer or the vet, unless the tumor is caused by a virus, but this is a different story. So basically, to become a neuropathology, it's quite a lengthy process. So I started in pathology like maybe 25, maybe more, 30 years ago. And I just like little by little concentrated more and more and more, just mainly in neuropathology. And basically I'm still learning quite a lot, but I'm now like pretty familiar with the disease in ruminants, and that's after 18 years like dedicating, you know, my career on that.
To filter out the questions from
is this feral species? I'm not sure, because maybe it referred to some.
Could you read?
Oh yes, that's great. Okay, so that's the one I just. So
is that the one you just?
Yeah.
Yep, that's fine. Just scroll down.
Addressed. So I have a question from Anonymous that asks, is this for all species? I'm not totally sure if this would have applied to a specific slide. Unfortunately, I cannot link the question to the slide, so I'm really sorry I cannot address this question. If it was about like the type of lesions and, you know, like overall the presentation I've given can apply to other species. I mean, you see like this type of like macroscopic and microscopic feature in many other animals. They may be slightly different from species to species, but generally are applicable to other species. And then I have a question from Ola Bizi Abdullah. Apologies if I don't pronounce your name well. Is there any difference in his histological views of ruminant brain? So histologically, as I was saying before, strictly, I mean, is if I'm put in front of a neuron with no other information, maybe quite tricky for me to know which species I'm in front of. So the ruminant brain, like histologically and at the cellular level, is very difficult to, to be distinguished just by looking at that. You have to know that which species and which animal you're looking at. So there are not specific feature just for that. But the anatomy, the anatomy of the brain can be slightly different from species to species. Then Younglan asked, can other diseases cause the brain to deform aside neuropathological diseases? So, well, that's an interesting question, because actually somehow the answer is yes, because if you have like a say a bone tumor in the skull, this would compress and deform the brain. It's not necessarily a disease that starts from the brain, but it affects the brain. I wouldn't know, like on the top of my head, any other, condition. I mean, of course, you have like external condition, like a trauma, which is not strictly a neuropathological disease as such, but it causes lesion in the brain. Then Anonymous asked the concentration of the formalin used to fix and preserve the brain. So this is, as I said, I have more detail if interested. Usually you say you use like formalin 10%, which is actually 40%. That's quite well. So basically you buy the formalin at 4%, if I recall well, and then you dilute it 10 times. So you call it formalin 10%, but in actual fact is 40%. However, there are, like SOP and I can circulate formula to prepare formalin if you want. Also, there are differences between countries, because if you prepare it with like distilled water, you then have to buffer it with other salt. If you prepare it with tap water in countries where the water is quite hard, you don't really need to do the buffering of the formalin. The important thing I want to state is that usually, like, the ratio of formalin versus the tissue is very important, and the ratio should be 1 to 10. Now, sometimes it's difficult because if you receive, if you think about a whale brain and you need, 10 times the formalin, you need almost a water bath to put the brain. But if you don't have enough formalin, if you don't put your brain in enough formalin, what you have to change it more frequently and possibly leave it longer. And in certain cases, we studied, especially for sea mammals that we also examine here, we have, we cut the brain through, then we put it back in formalin until basically the brain it's fixed. Another question from Anonymous, which is actually it is a good question. How are we gonna learn what has been taught? So basically, it speaks about my career somehow. So I can explain how I learned neuropathology. Usually, you have certainly to study neuroanatomy. So you have, before to get, an orientation, to be familiar with what the brain looks like and is like. Then you have to be also like familiar with the histology, have good books with you. But I think that the really, really crucial thing to learn neuropathology is to have good mentors, to have the privilege and chance to sit at the microscope and to observe cases and go through cases with some, you know, like knowledgeable, some experts on the field that shows you, like, and explain you what the feature are. I think this is, you know, like the way to how to do that, and that's why to become a, you know, like a better pathologist, the ideal is after the vet school, you can either go on and do a PhD, which is what I've done, because I wanted to be a research pathologist, even though I ended up being a diagnostician more than research pathologist. And then you can do a residency in different university where they're specialized on that. It usually lasts 3 years. And after the residency, most of the time, you know, you're requested to take an exam. There are different exams in different countries. The more well-known, respected and popular are the American board. Excuse me. And another one is the European board, but you know there are other areas where they provide residency on that, and basically then you are like a fellow. You know, you become a fellow of the society. I haven't done it. I didn't go through the residency or the exams, because in the position where I'm now, I didn't need it for a long time, even though I wanted to do it, but it was agreed that I didn't need it for the work I was doing because I was more research pathologist and I had a PhD. Now I regret a bit, I haven't done it. I would have liked to do it, but at this time I'm, you know, like it's never too late, but it's a bit not useful in my career to do that. But I would seriously advise you guys, anyone who wants to, you know, like go ahead and be a pathologist, to follow this either, you know, like this way of getting to any pathology.
Shall we say if we close the Q&A at like 1:15, because otherwise we're just because we're getting people asking a lot of questions.
Oh yeah. So I hear from yeah, no, I hear that you're getting a lot of questions. Unfortunately, we are getting short of time, but you can keep asking questions, and then I'll address them. You know, I'll answer them in the report you receive from Q&A questions. So you can keep asking, and I'll get back to you with, you know, like as well as I can.
I'll close the Q&A at 1:15, and I'll close the Q&A at 1:15, so no new questions can be asked after 1:15.
So also we'll close the Q&A at 1:15, so like roughly in 20 minutes, half an hour, and then after that you won't have the chance to do any question any longer. But well, still there is, you know, time to do that. You know, like I see some of the question. So basically like an anonymous asking about the PowerPoint presentation, and all of these, if I can share the lecture. So as I said, well, I'm just gonna maybe answer that. So as I said, you know, you'll be able to see this presentation recorded. I cannot provide at this stage with the PowerPoint presentation because I would have to ask permission to do that. From there are, you know, copyright images I used, and I would have to ask permission to colleagues, and I would have to ask permission to my institute to circulate the PowerPoint presentation. However, you're very welcome to go through, you know, to look at the presentation through the link that we'll be sending shortly after, once, you know, everything is all done. I may just pick another. I may just finish with Amethyst, who asked, what advice would you give to a vet sending samples for diagnosis to head your examination? I think this is a brilliant question. I didn't have time to address this question, but I have almost a whole seminar on how to, sample and why sample are important and how to do it. And would be very difficult, to answer this question. But if there is interest on that, I may be, available at a later stage to provide you with all this information with images, because sometimes it's difficult to explain, without any visual aid. So I think that I'm going to let me have a very, very quick look if there's some really okay, I'll take this one and then I close this one. Amino Abdul Salam, which asks, with respect to one of the slides on lissencephaly and cerebellar hypoplasia, is cerebellar hypoplasia fatal and can it be cured? So that's another, interesting and good question. So the hypoplasia, it really depends on how severe it is. Now, very strangely, and again, I would need another couple of hours to explain of that, but especially in animals, I see changes which are so extreme that I think a human, and sometimes though it applies even to human, but usually, in our case, in the case of us as human, we would have extremely severe symptoms, and we wouldn't, be surviving or set aside eating and do other things. But the changes are so extreme, but the animals still cope and still survive. And hypoplasia on its own doesn't kill the animal. Actually, there is, a case in cats, which is not now my area of expertise at the moment, but I used to do also neuropathology in small animals. And you can have cats being born through viral infection with hypoplastic cerebellum. They are a bit clumsy. They don't have this enormous sense of balance that cats usually have, but they can compensate for that. So this change is not always this vital. Of course, it depends on the level of the severity of the change, but also depends on whether the hypoplasia is associated with other lesions. So sometimes, this condition come together with other condition, and so in that case, the animal may die of the condition. It can be cured as such. I mean, I'm not aware, I'm not a clinician, and I'm not totally sure if there are new cures to improve the hypoplasia. Now, said that hypoplasia as such, because it happens during the development of the brain, and the brain and the nervous system is a stable organ. It doesn't reproduce, even though now new study have observed and reported that the brain can regenerate in part, but not as much as other organs. So there is not a huge chance for cure as such, but certainly there is a possibility for the animal to live a decently normal life if given the chance. And I think, as I said, that's us. I hope you enjoyed the presentation, and maybe I'll see you later on at some point. Thank you very much, and bye-bye.